Hi everybody, welcome to the second lecture in the experimental design and analysis section of the core skills modules. So everybody taking core skills modules across the School of Life Sciences, with the exception of pharmacology students, should be listening to this lecture. The first lecture in the series was delivered live, but this one and most of the others will be delivered in a pre-recorded fashion, so hopefully the fact that you're listening to me now means you found those pre-recorded lectures. Okay, so today's job is, we're going to look at exploring data. So this photograph on screen is an image that never ceases to amaze me. That's a photograph of the Martian landscape taken from one of the Mars rovers, and it sort of represents really the epitome of scientific exploration. The fact that scientists are still exploring the universe around us. And of course, in places much closer to home than Mars, we're still only beginning to scratch the surface of many terrifically exciting biological puzzles. And the starting point with any scientific exploration is collecting data and exploring those data. So that's my point, I suppose, with that picture. Off we go then. So the outline for today's lecture is, and it's going to be in three parts. We're going to deal with looking at the process of scientific inquiry, the scientific method that I described in the previous lecture. We're going to look at the first step of that process today, which involves collecting data and describing those data. And that will first of all lead us to think about the different types of data that we collect. So one of the objectives here is for you to understand those different kinds of data. You'll already probably be familiar with some of the distinctions I talk about today, so hopefully this is partly a refresher. Then we're going to move on to looking at descriptive statistics, which can be used to summarize data sets, because data sets in biology rapidly get very large and complex. So using descriptive statistics is a way of helping us to spot patterns in the data we've got, to summarize the data we've got. And in particular, we're going to look at the concepts of variability, so how variable are data, and central tendency, where is the middle of a data set, things like the mean, the median, and the mode. And then later on, we're going to look at visualizing data, so how we take data and explore them graphically. I'm not going to go into great detail here, but just a little bit of an outline of the main ways to graph your data in order to spot patterns. And that will bring us to the topic of confidence intervals and more generally error bars, which are used to graphically indicate confidence in estimates of the mean. And then the final section of the lecture is going to be about distributions, the different kinds of distributions that data fall into, what they look like, what sort of properties they have, and the consequences of that for our thinking about descriptive analysis and ultimately statistical analysis of hypotheses that we're going to come on to later in the course. So the first part then of this lecture is to look at collecting data, describing data, using words initially, recognizing the different types of data, and then using descriptive statistics. Okay, so in the first lecture I described this sort of simplified model for the way that scientific progress is made, and it is just a model, but it's a helpful way of looking at the whole of the scientific process from beginning to end. And what we're going to do over the next few lectures is focus in on different parts of this process to try to see what's going on. So today we're going to look at this initial phase in the scientific process where scientists observe the world around them, the biological world in our case, and frame questions about that world, which often are associated with a theory, a big idea. And that process actually is a really creative and exciting process, but it all starts with observation, and observation requires data. We need data in order to be able to quantify what's going on in the biological world. So broadly speaking, what happens is a scientist sees something interesting, an interesting pattern or phenomenon in the natural world, and says to themselves, well, what's that all about? In order to make any progress in framing an answer to that question, ultimately coming up with a hypothesis about what's going on, we need more than simply anecdotal observation. We need a systematic description, observation of a system in order to start to frame clear ideas about what's going on. So we're going to observe the system, and to do that, normally we would collect data. Notice, by the way, that all the way through this lecture and the others, I will be pedantically insisting that data is a plural. The word data is a plural word. So when I'm talking about data, I mean lots of data, lots of individual data points. The singular is data point, or if you want to be pedantic, you could call it datum. But that datum is very rarely used. And what you'll find in in popular science and in journalism and everyday life, people tend to use data as if it's a single noun, so the data is very interesting. But what you'll find in science is that you might well get criticised for that. So if you can possibly remember that data is a plural, it will help you, when you're writing essays and things. Okay, so let's look at data. What kinds of data are there? Well, data come in different shapes and forms. And recognising that is actually quite an important part of the process of going on to describe those data and then to make inferences from those data. So data can come in the form actually not of numbers, but of the names of things, what we call nominal data. So for example, perhaps a straightforward example is if you're looking at a group of ducks and you're looking to identify the sex of those ducks, they are going to be male or female. The data might come therefore in the form of a list of male, female, female, male, etc. Nominal data. But we use names for all kinds of categories in the life sciences. So for example, if you're trying to identify the genotypes of different individuals in a population, and you've got different genotypes perhaps giving rise to different phenotypes, then you might label those with, as I've done here, 4 different combinations of alleles. If you have a dominant allele and a recessive allele, this might ring bells from your A-level biology. We might denote the dominant allele capital A and the recessive allele little a, and so we might write down the genotype of a diploid individual in the following form: AA, or capital A, or capital A little a for a heterozygote, or little a for a homozygous recessive individual. Okay, so these are nominal data, and we could write them down for lots of individuals, and hey presto, we've got a data set. Nominal data are just one example, of a kind of variable that we call a categorical variable, so where the data are in categories rather than on a numerical scale, a continuous numerical scale. But sometimes categorical data are themselves sort of pseudo or semi-numerical. So anything that you can rank in the life sciences or elsewhere, like for example the answers to a questionnaire, you might be asking people for how strongly they're feeling some symptoms in a medical trial, and you want them to rate them from strongly disagree through to strongly agree, or something like that. Then what you're doing is you're asking the participant in that case to put the data into categories, but those categories have an inherent order. They can be ranked from first to last, in which case they are ordinal data. They're still categorical, like nominal data, but they now have a natural order, whereas nominal data, like the sex of ducks or the genotype of humans, they don't naturally fall into a natural order, whereas ordinal data do. And sometimes we actually take continuous numerical data and convert them into ordinal or ranked data for various purposes, as you'll see later in the course. But probably what we think of as typically as a kind of classical data set in science is a data set that is truly quantitative. In other words, where we are actually measuring something on a numerical scale. And quantitative data come in 2 main forms, where the data are arranged in discrete bins or where they are truly continuous. So if I, for example, count the number of bacterial colonies on a Petri dish, that count is quantitative. I'm counting how many there are there. It's numerical. I might have 3 colonies, or I might have 300, but you can't have every possible value of that numerical variable. I can't have -6 bacterial colonies, and I can't have 0.2 bacterial colonies. In fact, there are a number of discrete possibilities. I can have 0 or 1 or 2, but nothing in between, and I can't have negative. So this is a classical discrete variable, in a quantitative discrete variable. And what you'll see is very often nominal data are immediately converted into quantitative discrete data. So for example, if I'm looking at a lake with loads of ducks on it, I might well count how many males there are and how many females there are. And hey presto, I've gone from having a list of male, male, female, female to having a quantitative measure of the number of males and the number of females, but a quantitative measure that is discrete, because you can't have -3 ducks and you can't have 0.3 of a male duck. So what we're doing here is we're converting nominal data by counting frequencies into discrete quantitative data. So the other kind of quantitative data that we very commonly encounter in science is continuous data. So data that come on a truly continuous scale, either from -infinity to +infinity, or perhaps from 0 to +infinity, but where any value is possible within those bounds. So if I'm measuring the concentration of a chemical in solution, or I'm measuring how long it takes for something to happen, then those measurements can take any value on the scale that I'm using. And really the only constraint is the device that I use to make the measurement. So if I'm measuring time with a stopwatch, then typically the smallest unit of measurement on there is going to be a second. So I am still measuring the time in what could be considered to be discrete chunks, 1 second chunks, but that's only because of the instrument that I'm using to measure the quantity. The underlying quantity I'm trying to measure is truly continuous. That's not true when I'm counting the number of male ducks or the number of bacterial colonies on a petri dish. So hence this distinction between continuous and discrete variables, but the slight blurring of the lines there, because as biologists, as scientists, we are always using a device to make a measurement, and that means we sort of have to convert continuous data into what look more like discrete data as a result of the fact that we count time in seconds, or we count concentrations in whatever it might be, millimolar or something like that. So hence, there is some blurring of the lines between these different kinds of data, and that can also happen, for example, with ordinal data. Sometimes ordinal data, rank data, we use ranks as if they were a sort of really truly continuous measure of some process. You can even see examples where you have fractional ranks, this can occur where you have multiple individuals all with the same rank. You sometimes average the ranks and you end up with something being ranked at 0.3, even though that doesn't make a lot of sense in terms of the raw ordinal data. And for some forms of analysis, it's convenient to treat discrete data as if they were continuous, again, as we'll see later on. The key point here is that some kinds of analysis assume a particular distribution, and we'll see that at the end of today's lecture, and some of those distributions, the ones that we most easily can make use of mathematically, like the normal distribution, are actually continuous, and yet the data that we're collecting might be discrete or even ordinal. And sometimes it's convenient to treat ordinal or discrete data as if they were continuous, but the key thing is that you need to recognize that you're doing that because you're making some assumptions when you do that. And this all comes back to the kind of distribution that we're assuming, and we'll see lots more about that later on in the course. Okay, so let's assume we've got some data. What are we going to do with it? Well, the first thing you're likely to want to do, especially at the early phase of the scientific process, when you're really observing the natural world, trying to formulate questions about what's going on, trying to spot patterns, what you want to do is, well, you want to summarize the data and look for those patterns and quantify those patterns. And we do that with descriptive statistics, and we also do it with visualization of the data in the form of graphs and other kinds of illustrations. So let's look at some really simple examples of how we go from raw data to descriptive statistics. So this rather unpleasant shot of somebody sneezing on the slide is to demonstrate my example data set. In this case, I'm going to imagine that we have collected some categorical data on whether people are photic sneezers or not. So I don't know if you know about this phenomenon, but you probably do. Some people, if they look at a bright light or the sun, find that they are triggered to sneeze. Certainly this happens to me. I don't know if it happens to you. It's a kind of weird thing. I think I take some perverse pleasure in it. There's something quite nice about sneezing. But nobody really fully understands why it happens. There are some insights into what might be going on. The most popular theory is that the nerve that travels up from your nose to your brain to tell your brain that there might be something blocking your nose and you need to sneeze to try and get rid of it, that nerve goes very close to your optic nerve at the back of your eye, And when you experience a really strong change in the signal to your eye, so when you look at a bright light or particularly look close to the sun, you get a lot of neurological traffic going down the optic nerve, and there appears to be some crosstalk between that nerve and the adjacent sneeze detecting or blockage detecting nerve that's telling your brain about what's going on in the nose. So you end up with an accidental firing of the neuron from your nose, and the nerve from your nose, and hey presto, you sneeze. Okay, so let's imagine, and if we were in person, what I would do is actually ask you in the lecture theatre to find out. Let's imagine we sample people for whether they are or are not photic sneezers, because some people this just doesn't seem to happen to, and others it does. So imagine I've asked 10 people and I've found out that 4 of them sneeze and 6 of them don't. What can I say about that data set, a categorical data set where each individual is classified as either a non-sneezer N or a sneezer S? Well, seems blindingly obvious, but the very simplest thing I could probably say about that data set is how many individuals I've sampled, which in this case is 10. So in stats, and by extension in the sciences, in the life sciences, sample size is almost always referred to by the letter N, either capital N or lowercase n. And in this case, n equals 10. That's the sample size. That's easy enough. With discrete data like this, as I said a minute ago, we can immediately start to convert categorical data into discrete quantitative data. So here we've looked at the frequency of sneezes by counting up the number of S's in my data set, and you can see that there are 4, so the frequency of sneezes in that data set is 4. And expressing that a different way, we could say, well, what's the probability of an individual in that population being a sneezer if I draw that one of the individuals out at random? So if I pick a data point at random from the data set, there's a 0.4 chance, or 40% chance, of picking a sneezer. So the probability of being a sneezer is 0.4. And you can mess around with this. You can say, well, what's the probability of not being a sneezer, which is obviously 0.6. Okay, so that's categorical data, and we're quite restricted with that kind of data set in what we can say. But as soon as we have quantitative data, we can say a little bit more using descriptive statistics. So let's look at a real example this time. So in the photograph here, you can see this Land Rover, and next to the Land Rover is just in the background my student Jess, and this is a PhD student who was working out in Botswana. And what she's actually doing there is counting hyena feces. I know it's not a particularly glamorous job, but somebody's got to do it. So she was actually doing a PhD looking at how hyenas communicate with other large carnivores like lions, which is the other animal in this shot, at latrines. So hyenas defecate in latrines, big piles of poo basically, and wee, and lots of other interesting olfactory cues. And those latrines are really important for hyenas in order to be able to communicate with each other and also with other species. So as part of the project, Jess was counting the number of what we call scat or poos at each latrine over a long period of time to try to understand the dynamics of this communication network. And here's an example of the sort of data set that she generated. We've got 20 different latrines here, and at each latrine she's counted how many scat are present. And some latrines go basically the scat disappear after a period of time, and so you end up with 0. But some of the latrines are quite large, so there's one there that has 11 scat in it. So the sample size here is 20, so that's the first descriptive statistic that we can provide. But because we've now got quantitative data, we've quantified the number of scat, we can do slightly more sophisticated things that you'll be pretty familiar with. And an obvious thing that we might want to do is to describe the sort of central tendency of this data set. In other words, what does a typical latrine look like? What's the average number of feces that we find at a hyena latrine? And hopefully you're familiar with how to do that. It's pretty straightforward, calculating the average. But mathematically, when you write it down, it already, even though it's a really simple thing to do, starts to look quite imposing. So this is the equation for the mean. In stats, we typically indicate the mean of a sample using a little bar above the letter that we're using for the sample. So here we've got x bar, meaning the mean value of x, where x is the number of poos per latrine. And that's calculated pretty straightforwardly from the sum of all of the individual values of x from the first observation up to the last observation. And in a data set where you know you have n data, in this case 20, the last observation is the nth observation or the 20th observation. If you're not familiar with this Greek letter sigma, it means sum or add up. So we add up all the values of x, and the little bit at the bottom here, I equals 1, tells you where to start summing, and the bit at the top tells you where to end. So here we're adding up from the first to the last data point, all of the data. And then we divide through by the sample size. And that's how we calculate an average. Everybody knows that, hopefully. So pretty straightforward. In this case, the answer is 3 feces or scat per latrine is the average. And that gives us an immediate idea. Instead of being bewildered by a great long set of data, we have an immediate single simple summary of what the data are like. And what they're like is there are typically, on average, about 3 feces per latrine. But as you'll be aware, there are other ways to express the central tendency or describe the central tendency of a data set. So we take the same data, and if we put them in rank order, as I have done here, from the least scat to the most scat, and look for the middle observation, and if there's an even number of data points, then we have to take the average of the 2 middle observations, because obviously if you've got 20 data, then you're going to have 2 numbers in the middle. Then you get what's called the median, and the median is a different way of expressing the central tendency of a data set. And as you can see here, the median is 2 scat as opposed to 3 for the mean. So the median and the mean can be different, and that tells you, the difference tells you actually something about the distribution of the data. In this case, we've got a few latrines with quite a lot of scat, 6, 7 and 11, and lots of latrines with only 0, 1 or 2. So we've got quite a skewed distribution actually, weighted towards 0 with occasional observations on the right hand side of the distribution. We'll look at distributions later on. And that's why the median, the middle value, is lower than the mean value, because the mean is pulled up by a few large observations, 6, 7, 11. And to the trained scientist, that kind of information, the subtle difference between the mean and the median, is informative. It tells you immediately something about the whole data set. In this case, it tells us about the sort of shape of the data set. How dominated is it by low values or high values? Okay, and you probably realise there is a third way to express central tendency, and that is the mode. And that's just the most common observation, the most common number in the data set, which in this case is 2. If I arrange the data, you can see that there are 5 latrines with 2 scats. And that's often also different from the mean and the median. In this case, it happens to be the same as the median. And again, it tells us something slightly different about the central tendency in that data set. So all of these very simple descriptive statistics, or what are sometimes called summary statistics, help us to describe the central tendency of the data. Okay, so that's the end of the part 1 of this lecture. In the next part, we're going to go on to look at not summarising central tendency, but summarising the variability of a data set using descriptive stats, before we go on to look at using graphs.